All right, so we have some lab presentations on Monday. If you are presenting, make sure you get your presentation together. Assembly. Let's see here. I have the lecture quizzes. Let's see here, the other flagellates. I think that deadline is today. When I did the Nevozoa, I think I did Thursday. Avoid the possibility of perhaps university cancels classes on Friday. Who knows? And there's some issues that happen. I was just talking to some staff that aren't very happy about the decision. We'll go on as if we were having class. And then if we do, next Friday, I'm out of town. There's no class. I think what we might do is just call it a day. Take that time to review, study, and so forth. All right? All right, let's move on. So we finished the Nevozoa. Last part are the alveolates. This is the phylum Apicomplexa. This is probably the most intense part of the class. If you thought it was intense already, the number of new definitions and terms is just going to increase. I'd like to apologize, but I really can't. We need those names. We need those terms. So. Best thing I could do is, best thing I suggest is, as you go through this, you have the definitions, in your notes, and then maybe day after start assembling just a sheet with just definitions and terminology and so forth. All right. So we're in basically the large group, the alveolata. Right? It's above the phyla level. Above the phyla. And this group is named because they have cortical sacs, cortical alveoli, hence the name alveolata. What are these things? Well, you would typically think of these sacs as saying we have our membrane, and then you would think of the sacs as being saccular. But that's not necessarily what they look like. Instead, what these things are they're more flattened right underneath that membrane. All right, that's what your alveoli. The purpose of these is that it helps to support the cell membrane, provides more structure to the cell membrane. All right, so that's where it got its names, these alveoli. Members of this group also possess micropores. And now you do have these small openings in the plasma membrane that small molecules can go into and then be absorbed by the individual. Our individuals have mitochondria with tubular cristae. So we've seen those tubular cristae before, right? And this group includes the dinoflagellates, the ciliates, and the Apicomplexa. So the alveolata is a really large group. That's our introduction to the alveolata. We're going to focus on the Apicomplexa. Ready? All right, so the phylum Apicomplexa has a large number of species. Estimates just over 5,000. Over 5,000. Can't really give you a real definitive answer because a lot of times more apicomplexes are found as additional organisms are searched for these apicomplexes. It's really easy to find, nematodes and tapeworms and flatworms and canthocephalans and so forth in an intestine. It's a bit harder to screen those things for apicomplexes. So as people have looked, for the apicomplexans, they've started to find them in different species. Some of our apicomplexans are important for human health. For example, malaria, that's an apicomplexan. Toxoplasma, that's an apicomplexan. And those are two species we're going to talk about. Others really don't bother us. They're going to be parasites in other organisms. This phylum, Apicomplexa, is named because all members have what's called an apical complex. This apical complex is a collection of structures at the anterior end that aids in tissue penetration or cell penetration, I should say. So they all possess this apical complex at that anterior end. The function of this apical complex is to enter a cell, penetrate a cell. So what makes up the apical complex? There's four things that we're going to talk about. First one is a polar ring. It's right at the top. This is one or more rings of anchor microtubules that support the rest of the apical complex. Think of it as kind of the scaffold that holds everything else together. Right? It could be a single ring, it can be multiple rings, and that kind of contains and holds all of the other structures. Next thing are the conoids. Conoids are a number of coiled microtubules that exist as fibrillar cones inside the polar ring. These may aid in penetration of a cell. So we've got our polar ring, and parts of it are cut out. But you have to know the polar ring kind of circumnavigates or circumscribes, goes around that interior end. So they kind of blocked off, or they eliminated that front. So that way you can see these coiled microtubules that are the conoids. It's thought that it aids in penetration because a lot of times when you think about proteins that help in penetration of cells, like in virus entry and so forth, they've got proteins that are helical, and then they make contact, and then when they uncoil, they shoot forward and through the membrane, and then causes fusion of the membranes to get inside. So that's kind of why they think that it goes into this penetration. It resembles some of these coiled proteins. Ready? Polar ring, conoids, and then we have rock trees. These are here in our figure. Rock trees are tubular sacs. Used in proteolytic entry. So tubes, sacs, it's all about proteolytic entry. They're going to secrete some substance that aids in the penetration of our cell membrane. And then the micronemes are elongated tubular organelles, possibly used in the penetration of cells. So the micronemes are kind of these black things that are scattered throughout. We'll talk about, we know the sequence of entry for Plasmodium, which is malaria. Right? And when we get to that, we'll use malaria as an example of how the things in the apical complex function to get that parasite into that cell. All right. These are going to be there, but then they disappear once our parasite gets inside the cell. Once they get inside the cell, they have no need to maintain any of these structures. So you're not really going to see these in every single life cycle stage. It's only going to be found in those life cycle stages that are going to penetrate into a cell. This diagram is a really good diagram of an apicomplexan, but it's way too big. Our ability to see these things is basically, yep, that looks like that could be a apicomplexan, and that's about it. All right, we'll get sometimes a nucleus will stain, and that helps. Sometimes you'll get staining at the apical complex, so probably the rock trees are staining and so forth. So in terms of seeing all of the details, there's no chance we're going to see it.
I was just going to ask, like, then we do be able to know the structure of, like, much stronger microscope?
Yeah, SEMs. SEMs. Transmission electron microscopes is how we know structures. So this is going to be a good one where you might have, a diagram. This is representative apicomplex. What's this region? What are these structures? Or whatever. So this is a schematic of apical complex of Toxoplasma gondii. Just so you can see that we have these apical rings, polar rings. There's 3 of them. You've got your conoids that are coiled and circular. You've got some subfollicular microtubules that run underneath the actual cell membrane that provides structures for that organism. But this kind of lays out where the apical rings are, polar rings, and how the conoids are there. And then in the interior, that's where you're going to have your ruptures and your microtubules. Present that for completion. All right. So micropores, that's part of our alveolata. We're going to find these micropores in sporozoites, merozoites, and trophozoite stage. So they're present, and here's it is what it is. It's a small pore in the membrane. It functions similarly to a cytostome in terms of endocytosis and stuff, but we find it in various stages. And these stage names, sporozoite, merozoite, trophozoites, will define those. We will be defined. Members of this group reproduce sexually and asexually. And there's a bit of clarification here. So we're not dealing with ovaries and testes and so forth. What we're dealing with is chromosome number. So if we have sexual reproduction, we're going to have the fusion of gametes to double our chromosome numbers, and then we're going to have some form of genetic reduction, chromosomal reduction, meaning meiosis happening. It's likely that most of the life cycles involve haploid individuals, and then the diploid stage is just a very brief time point where you have fusion of gametes, you can have recombination occurring, produce increased diversity, and then you have meiosis that happens fairly quickly after that. Or all of that recombination is occurring during meiosis, which would happen fairly quickly. All right, so we're going to see sexual and asexual stages in the life cycles. And also, many of these species have oocysts. All right, oocysts are essentially spores. And again, we'll see these terms, we'll define more of these terms. But the oocysts have cell walls. They have a thick outer covering because this is usually a transmission stage, and you need that wall for protection from the environment. Ready? Yeah. All right, so in this phylum, we have 2 classes, Conoidacida and Aconoidacida. Where do we get the names? Well, conoids are in our apical complex. The Conoidacida is those classes where the conoids are present. The Aconoidacida, A means not, right? Conoids are generally absent. It's not as clear cut, but it is what it is. It is what it is. So the Conoidacida, conoids are present. We've got 2 different subclasses that we're going to talk about. We've got the subclass Gregorinacina. These are the Gregorins. And then we have the subclass Coccidiacina, which are the Coccidians. We're going to talk about both of these. And there's more subclasses, but these are the ones we're going to talk about. The Aconoidacida, we've got 2 orders that we're going to talk about. We're going to talk about the order Hemosporida, which is Plasmodium. And then we're going to talk about the order Pyroplasmorida, which is Babesia and Phylaria. This is kind of where we're going. classes in our Apicomplexa. Ready? All right, so we're going to start in the class Conoidacida. Order, Eugregor in order. These are Gregorins. Generic names are Gregorins. These are Gregorin parasites that infect invertebrates, primarily annelids and arthropods. So annelids are segmented worms, and then our arthropods are insects. Members of this group are going to infect the body cavity, the intestines, or the reproductive system. Usually you find them in the intestinal tract, or at least their life cycle. One of the life cycles is reproductive sac. The other life cycle is in the reproductive system. Transmission among the Gregorins is by consumption of our oocysts, which are the spores. And we're going to point out here that merogeny is absent. So we go back to our protozoan introduction. We talked about merogeny, right? That's multiple fission that results or produces merozoans. So what's multiple fission again? Now we're kind of introduced. Now we're encountering multiple fission. Yeah, so we're going to have multiple nuclear divisions happening before that cytokinesis event completes, before that first one. There's really only one cytokinesis event. That one event is going to produce many daughter cells. So in the Gregorins, merogeny is absent. We're not going to see merogeny. It won't produce any merozoites, which is a good thing. There's no merozoites in the life cycle stage. Okay. All right, so there's two suborders. We're going to talk about both. The first suborder is suborder Aseptoturina. And the second suborder is Septoturina. So again, here's our term Septoturina, Aseptoturina. Right? One of them is going to have a septum, the other is not. What is this? I mean, you'll see some images to provide clarity. But our gamete will either be a single body, it's our Aseptoturina, or the gamete will consist in two parts with a septum between the two. Hence the Septoturina. You've got a septum or you don't have a septum. Our representative examples are Monocystis lumbrici. That's our Aseptoturina. We're going to dissect a nightcrawler to look at these guys. And we're going to do that on Monday. And then the other one is Gregorina cuneata. We've got Gregorin slides. I have to pull the strength. Keep forgetting to go get it. But we have some prepared slides for those guys. If we wanted to find our own, I'm sure we can. I'm sure we could find them. We just have to pick up some insects. All right. That's a general. We're going to talk about Monocystis lumbrici. Ready? All right, so Monocystis. This is a parasite of the seminal vesicles of earthworms. And this is a pretty good drawing because it kind of gives you an idea of where everything is. All right, so if you've had zoology, I think you dissected one, right? Was it preserved? Was it preserved? Oh, you can't remember? Anything that was preserved, did you get to see any colors or anything? It all looked like the same color, right? Hard to tell. What's that? Sassafras, right? Bunch of food. Yeah, okay. We're not doing that. We've got live night crawlers that we're going to dissect. You're going to see the colors. It's going to be very obvious to see where the seminal receptacles are. So, or seminal vesicles. So, anterior end, that's our earthworm. You get the mouth, all right, and then going down to segment 7 and 9, that's where our main seminal receptacles are. And we're going to be able to see the hearts and the dorsal heart that are on these guys. You'll see some blood from the worm and everything. But these worms are going to reside, or the parasite's going to be in our seminal vesicles. So what we're going to do is we're going to dissect out the seminal vesicles, put it on a slide, add a cover slip, squash them, and then really look to find all of these different life cycle stages. All right? So let us go through the life cycle. I'll say, I think COVID that year was the first time that we actually diagrammed the life cycle in lecture. All the other ones, I kind of I verbalized it in the lab manual, and then really it was kind of meant to go try to find out the life cycle yourself. And I think by diagramming it in lecture, it makes things a whole lot clearer. So we're going to use all of these terms that are up here. Is that a nice piece of chalk? That big thing. Where are we going to start? Let's start down here. All right, so we are going to start with a mature spore. All right. Our mature spore will have 8 sporozoites. 8 sporozoites. This will be out in the environment, and this mature spore will be consumed by our earthworm. All right? So then once you get into the gut, the spore will open, releasing the sporozoites. Now, we already said, all right, the primary site of infection are these seminal vesicles. So what's going to happen is that these sporozoites are going to penetrate the gut and migrate to the testes. And they're going to do so by the heart. So they're going to penetrate out of the gut, get into the circulatory system, go to the heart, and then find their way to the testes, where we now have our sporozoites. All right. At this point, our sporozoite is going to look something like that. Pretty small. It's going to be hard to identify and hard to see. All right. Then these sporozoites in the testes are going to penetrate these cells that ultimately produce the sperm. So what we'll call sperm-forming cells. So they will penetrate this cell and they will transform into a trophozoite. They're on these sperm-forming cells, and what they're going to do is feed on these developing sperm. And as they feed, they will get bigger and bigger and bigger. And in some cases, we'll actually see some where now our trophozoite's much larger, but it's going to look like it's hairy, or have a bunch of flagella, because that's basically the tails of the sperm of our earthworm. Earthworms. All Alright, so they get enlarged, still trophozoites, and then these sperm-forming cells are going to release, and now we will be a trophozoite in the lumen of the seminal vesicle. So as it matures, as it consumes that sperm-forming cell and gets larger and larger and larger, it will then transform. It will transform into a gamete, but there are two types. Gamete. There's a mating type 1, and then there's a mating type 2. We don't know which is which. But we're still in the seminal vesicle. All right, so we have these two gametes. All right, they are going to look fairly large. And it's gonna be kind of what they look like. Sometimes you'll see your nucleus and so forth. All right, but we know there's two types, and in order for our sexual reproduction to occur, you have to have fusion of these two types. And that process is called syzygy. Memorize that word, so when you play Scrabble, you can get loads of points. All right, S-Y-Z-Y-G-Y. All big points. Probably like only 4 Y's in that game. All right, but before that happens, they're going to attach to the epithelium. They're going to attach close to each other, and then they're going to pair up in syzygy. So now we have paired gametes. And these things, they will be attached. They will look something like that. And again, fairly large. Notice how I did the sporozoa pretty small. Or I tried to demonstrate it was pretty small. So that's our paired gametes. So if you're counting, we're at like our 2n number. Right. We've paired, we're fusing. All right, so we're paired, pairing of the gametes, that's syzygy. And then what we will have is fusion, ultimate fusion of the cell membrane, of the cytoplasm. And then cyst wall formation. And once we have that, now we are called a gametocyst. So we're going to have this, our paired gametes, and we'll have a cyst wall inside of it. That is called a gametocyst. Next thing that'll happen is we will have gametogamy. Gametogamy, which is the production of our gametes. So now we still have a gametocyst. Instead, they were gametes. So I guess we haven't completely fused. Missed that part. But we've got our gametes. Now, how they look is basically you're going to have your cyst, and there's not going to be a whole lot of structure inside of it. Because what's going to happen is that one of these will form the male gametes, the other one will form the female gametes. but we can't tell which one's going to happen. We don't know which one's going to happen. And then, once we have these gametes inside, we will have fusion of the gametes. There's our two walls. And then cyst wall formation. So now we will have a gametocyst with oocysts. So you have the fusion. They lay down a cyst wall to produce our oocysts, which is our spores. So you're going to have this gametocyst now with all of our spores inside it, etc. And the age, you'll be able to tell different ages, because the more full these things are, the more mature the gametocyst is. More mature. So each of these would be an oocyst. And then we are going to have our sporulation and then sporogamy. So sporulation is our production of say spores, but it's not. Sporulation is basically the meiosis event that happens. So we're going to have our meiosis event to produce our sporozoites, which then undergo sporogamy to produce the different sporulation, I should say. I have sporulation and division. This is probably best. Just say it. Sporogamy is both. Because we see that in our life cycles. The sporogamy event is the genetic reduction and then division to produce our sporozoites. So now we have our gametocysts with the oocysts containing the sporozoites. All right. Now, we're inside this gametocyst. What's going to happen is that this is going to have to rupture at some point. Okay? This is going to have to rupture. It can rupture before it gets out or after it gets out. But it gets released through the male genital cord. So we're going to have a rupture before or after leaving the host. All right, so now we have our environment, our host, earthworms, neck crawlers. I group them together, but you say earthworms. Don't say worms. That could be what? Flatworm, roundworm, tapeworm, annelids, oligochaetes, earthworms, neck crawlers. All right, and then you get consumed one of these mature spores with those 8 sporozoites. Couple things. Our worm can also consume a gametocyst. So if the entire gametocyst comes out, our worm could consume a gametocyst, and then that could rupture inside the intestine. But we still have to have sporozoites in the oocyst in order for a worm to become infected. Got that possibility. Release is the other thing. What eats earthworms? What's that? Birds. So our escape could also occur when the worm gets eaten by a bird or a mammal, because these gametocysts can travel through the gut. And that term for gametes where you can't distinguish between the male and female, anisogametes. We don't know which one's type 1, which one's type 2. We don't know. They look identical. All right. So, life cycle stages that we'll be able to see in the lab. We may find one of these trophozoites that have the sperm tails on them. We'll probably find these, could be either a trophozoite or a gamete. When we pull them out and crush them, I do think we end up causing some of them to detach. So it's going to be difficult to really identify trophozoite versus a gamete. Definitely if we could see the flagella on here, we know it's a trophozoite. Gametes will not have it. We possibly could see this. I know we will see this. So really the difference will be, can we see the cyst wall or not? All right, and then we will see various stages of our gametocyst. Some where you'll actually see the oocyst inside of them, some where you only have a couple because they tend to form on the periphery and then it fills up the middle. So it kind of works from the outside in. And of course, we're going to look at the spores, which I didn't really draw what a spore looks like. Looks something like that. It's got these 8 small sporozoites. We can see. Do we need oil immersion? No, we don't. We're going to see a lot of this stuff at 4x and 10x. Probably go up to 40x to really take a look at some of these spores. Questions? A bit longer, but just know the pattern. All right, sporozoite's going to transform into a trophozoites. Trophozoites grow, get released, they transform to a gamete, they attach. Syzygy happens where they pair. Once they pair, they lay down the cyst wall, then it undergoes gametogeny to produce your gametes. And at this point, once they have the cyst wall, it's called a gametocyst. Gametocyst is going to contain our gametes, and then the gametes, and then the oocysts. So the oocyst will be inside the gametocyst. Oocysts will have sporogony happen inside of them, producing our 8 sporozoites. So it's just kind of a sequence of events. So, got these terms up here. The gamete. That's our life cycle stage that undergoes gametogamy to produce our gametes. Syzygy. That's our pairing of gametes. Anisogametes. Situation where the gametes are indistinguishable. That means there's no evidence of either micro or macro gametes. So go back to botany. Some of you had botany, right? Those of you who haven't, you will learn micro and macro gametes. Micro and macro gametocytes. Right? No evidence of that here. They're indistinguishable. That's an isogametes. And then the oocyst It's a cystic form in the Apicomplexa containing the zygote. Could be called a spore when it contains sporozoites. And I have that there because we're going to see an oocyst and some other life cycles as well. So here's some of the life cycle stages. Spore, sporozoites inside of them. We'll see the spore wall. We'll see it. It's going to be hard to really identify the sporozoites sometimes. We'll get lucky and see evidence of sporozoites. Again, these things are going to be pretty small. You can see the spores at 10x. You see them better at 40x, but it's still pretty small. Here's a situation where you've got your trophozoite. There's all your sperm tails. So I know this is a sporozoite. This is a gametocyst. I can see the cyst wall, and I can see each of the individual gametes inside of it. And note the size. They're going to be visible. I've never once had a night crawler not be infected. I should knock on wood. This will be the year that everything's uninfected. But yeah, it only takes one to get in that farm, and then everyone's going to be infected. So this is a pretty easy one to find. All right, questions?
Are there any adverse effects on the earthworms?
On the what kind of worms?
On the earthworms?
Earthworms? Not that we know of. Guess I didn't do that. Yeah, I have a Gregorian pathology, because we'll talk about Gregorina as well. Pathology on the insect host, we have to assume that there is some sort of negative aspect to it. I mean, earthworms losing some sperm-forming cells, but it doesn't seem like it negatively affects them. I think you could look at like population level responses. Is the growth rate inhibited for infected versus uninfected? And then trying to find uninfected ones that are pretty hard. I have not pulled out any, I have not gone out and tried to capture my own worms to look at them yet. All these night crawlers, I've always bought them at Walmart. Every single one's infected. So, I can't imagine it's too bad.
So they can still sexually reproduce, the worms can, even if they're infected?
Mm-hmm. Yep. They can still sexually reproduce. Any other questions? We're not gonna. We'll take a break. We'll talk about Gregorina on Friday, unless things change. All right. So we'll look at this. We'll try to find as many life cycle stages as we can. All right. I would sometime between now and Monday, I would sit down and try to map out this life cycle and the pattern, the order in which it occurs, and maybe just briefly draw out some of these stages, so just so you have an idea of what you're looking for. Again, we're going to be able to see this stuff at 4x. Usually, we spend most of our time at 10x. And then we'll zoom into 40x to take a look a little bit more closely at the spores. All right, let's call it a day.